All right. So now a panel that we have every year, which is always one of our most exciting panels because it's such a fast-paced area, and that's oncology. So let me introduce the moderator, Dr. Najat Khan, and then Najat, you can introduce the panel. So Najat is Chief R&D and Commercial Officer at Recursion. She brings deep expertise across biological, chemical, medical, and data sciences, along with business leadership focused on delivering patient and business value. Previously, she was Chief Data Scientific Officer and Global Head of Strategy and Portfolio at J&J. Dr. Khan has pioneered large-scale use of data science and AI, building a top-tier diverse team and advancing innovation at the intersection of healthcare and technology. At J&J, she co-chaired the Data Science Council, promoting company-wide adoption of data science, best practices in AI, and ethics, as well as talent and development. So Najat, I'll hand it over to you.
Thank you. It's great to be here today. Thank you, everyone, for making the time. I have such an esteemed set of panelists that I am not going to introduce them because they don't need any introduction. Instead, I'm going to spend the time to give you a little bit of a 1-minute intro, and then we'll jump to questions. When we did prep sessions for this panel, we did it across 2 meetings, and they all went over. So being pithy is of the essence. All right. So look, oncology continues to lead the industry in terms of investment, innovation. It's one of the top R&D areas, spend areas for nearly every pharma and biotech company. But it also is coming up with a lot of competitiveness, which is now becoming more global, such as the innovation from China. What's exciting is that we're entering a new era. And I know we say that every year, but we'll talk through it. Especially in terms of what is going to be, as Sam Waksal has coined, paradigm busters, driven by the advances in targeted therapies, IO, cell gene therapy, and so forth. But it's not just that. We're also seeing innovation in how we interrogate tumors, ctDNA to AI-based pathology, and transcriptomic. Precision medicine is evolving beyond genomics into multi-omic, spatial, and temporal insight. And again, it's oncology that's leading the way. So the leaders with me today represent a spectrum of innovation, from drugging the undruggable and advancing kinase and immunomodulating therapies to engineering next-gen cell and gene platform, to driving clinical development across oncology and beyond. With more tools and a lot more noise comes more pressure. How do we raise the bar on what we advance, and how do we spot these differentiated therapies earlier? So I'm going to start with my first question, which is the evolution of the opportunity. Oncology looks very different from 10 years ago. What are the current paradigm busters that each of you are excited about? And if you want to, maybe add a flavor of how it was different from 10 years ago. Sam?
She called on me because I'm the only one that's been around so long that I remember 10 years ago as if it were yesterday. So things have changed in oncology, and if we think about it, 20 years ago, or when Stelios started 40 years ago, there were things that were really different. We were barely able to understand how to drug tyrosine kinases, and people were just beginning to use something other than chemotherapy. And in fact, that came much later. And more importantly, even later, after 100 years of work, we began to understand how to harness the immune system. And that was a really big deal. It was a game changer. I mean, I really worked on making antibodies to tyrosine kinase receptors, things like EGF receptor with Erbitox, things like Cyramza with anti-VEGFR2. So those antibodies were really turning off signaling agents. But then we began to use antibodies in a different way, in a really different way, and we began to modulate the immune system, and that was a big deal. It was such a big deal that it literally changed how everyone was doing work. But it not only changed things clinically, it invigorated the biotech industry. And everybody began doing things that were not paradigm busters. They began trying to copy what Medarex and what Merck and others had done. Bristol Myers, of course, bought Medarex. So that changed a lot of the industry. But then bigger changes, antibody drug conjugates, the work that Antoine Yver and Jose Baselga and others did to move Enhertu forward became game changers. For the first time, instead of needing millions of molecules on the surface of cancer cells, Enhertu was able to kill cancer cells if they had 4 HER2 molecules. And now that's being used first line. So that created a surge again. You know, the biotech industry, Aristotle called human beings zoan politikos, political animals, not because of anything to do with politics. He believed we ran in herds. And the venture community is an example of that. If there's a company that gets exciting work in an area, or 2 companies like Lilly and Novo for GLP-1, suddenly there'll be 50 companies that are doing the same thing. So we run in herds. But in the cancer area, there have been some cool drugs, and not only ADCs, but CAR Ts, and now we use radionucleotides to bind to small molecules or to antibodies, and cancer keeps moving forward. And of all the areas of disease need, cancer is a great area because we can really do things for unmet medical needs. There are still cancers that are very difficult to deal with. Pancreatic cancer is still a nightmare because of fibrotic tissue, because of other things. And there are other cancers, glioblastomas and other ones that are still very difficult to treat. But we're learning.
So Sam, I might move it to Paul to
I can
maybe
talk
talk
for a minute.
to share a little bit more of looking forward for each of you. What are the areas that you're most excited about?
I'll just start by saying that I completely agree with Sam's point about the need to change paradigms, and that there's one big echo chamber and herd mentality. And this is a deep problem from a philosophy of science perspective, right? And there are beautiful books about this, like the structure of scientific revolutions that everybody should read, I think. Because we are wasting, especially a lot of money in every field, including oncology, because we try to have another PD-1, or now we have how many PD-1 combined with VEGFs, right? More than 30. There's a huge waste of resources. So the question is, how can we start to see the future, to your question, in a more effective way? I think the goal should be led by the patient unmet needs. And unmet needs are clearly defined by some of the examples already, like glioblastoma. My company, I'll speak about this in a second, how we are trying to do that, has a program in recurrent glioblastoma. Probably the worst of all the glioblastoma patients are those who have already failed gross total neurosurgery, right? Gross total resection, and chemotherapy, and radiotherapy. And we have a program that seems to work. And if you talk to investors, the initial response is, yeah, we don't do glioblastoma, or pharma companies alike, because this has been a graveyard for the industry. But it's always like that, because everything will always fail until somebody figures out how to do it. So I think we really need to start to look at the data. So start with the big unmet need. Recurrent glioblastoma is a great example. Pancreatic cancer is another great example, where we do have a program that looks extremely encouraging based on a small randomized clinical trial where we look at the hardest endpoint, overall survival. Same is true for therapy-resistant non-small cell lung cancer. But also, on the other side of the spectrum, if you think about prostate cancer, which is the second most common cause of cancer in men in the US and in many other parts of the world. And number one would be all the skin cancers, including innocent skin cancers grouped together. So big unmet need. Many people have started to think, well, most men will die with prostate cancer because they get a heart attack or another reason why they die. But it's also true at the same time that this is the most important cause of mortality due to cancer in men. These things are actually not mutually exclusive. So there are huge misunderstandings about these concepts. And I would say the biggest unmet need in prostate cancer is probably not in advanced disease, but in early localized disease, where we've not seen any successful phase 3 program in more than 20 years. So I'm very pleased that with our new approach of viral immunotherapies, which is actually not an oncolytic virus that we test as a lead compound, it's a combination almost of in vivo gene therapy and in situ immunization, that we have reached actually the primary endpoint of disease-free survival, basically cure, in patients with early localized prostate cancer. We've seen positive data in prostate cancer. We've seen in pancreatic cancer. We've seen positive data in completely therapy-resistant lung cancer with a whole new approach. So I do not only share this to highlight actually that we have some very interesting data that not everybody may be aware of. All of this came actually the last 3 to 4 months, but also to reinforce Sam's point about the need for paradigm shifts. I'll make one very additional quick comment and I'll leave time for the others, is that I think the future will be increasingly focused on combination therapies, which have of course been around for a long time in oncology and also in other fields. But we need to be really rational. I'm also an immunologist, so my bias is also that almost everything is, in the end, touched by the immune system. And even in the patients who currently do not respond to immune checkpoint inhibitors, the question will be, how can you convert non-responders into responders? This is possible by design, by combining with new approaches. The same is true for CAR T cells, which have been hugely successful and transformative, as we all know, in hematology, in liquid tumors, but not so, generally speaking, in solid tumors. What is the big difference between a liquid tumor and a solid tumor? It's the absence of the tumor microenvironment. So how can we change the tumor microenvironment so that you can actually attract CAR T cells into, for example, pancreatic tumors by breaking down the extracellular matrix and then allowing them to enter the site of the tumor, and then after entering the tumor microenvironment, how do you make sure that they stay happy and functional and don't get exhausted from an immunological perspective? So these things can be done by design, and I give this as examples of the strong need for combination therapies. Thank you.
Steve?
I'll try to be brief. So I'll go back and just think a little bit about what we started this, some of the topic this morning. It's like value creation and value recognition, right? And we're in this era where value recognition is coming from really just a specialty pharma, same target, multiple approaches. And it's not really necessarily value creation. And true value creation sometimes takes longer to recognize. And that will come through innovation. And the thing that I'm frustrated by is one, the frequency with which we go after the same targets with new modalities. 2 is how frequently I'm wrong about different things, and that includes things like the simplicity of, well, maybe we could cure things just by turning on the immune system and it'll work everywhere. But what makes me optimistic is I feel like we're at a moment where we now have the tools to really figure out how much of these different cancers are actually just syndromes, and what are the true diseases. And that will allow us to really partition the patients and design the right therapies, and they will, I agree, be almost certainly multimodality and multi-targeted. But we'll figure out that these are actually distinct diseases. It's not pancreatic cancer, right? As we know that in a number of these singular genetic point mutations and things like that. But I do feel that's what the part of it where I feel value creation will come, and where we will really find some new therapies.
All right, thank you. So I will be very brief and hopefully provocative. My view is nothing has really changed in the last decade, and I doubt a lot will change in the next decade, okay? New targets, new modalities, new ways of looking at things are important. We're in the p-value business. That means we have to run large, randomized, global pivotal trials, and we test hypotheses in that setting. Okay, that's where the action's at. That's how you get into the game of helping patients, right? My job, our job at Exelixis is to improve the standard of care for cancer patients every single day. And if we can't do that in a pivotal trial with the right p-value in the right population, in a large enough population to move the needle for patients and shareholders, then we're not doing our jobs. So that's where the action's at, right? Sam mentioned in HER2. Well, that's interesting. That I view as an incremental improvement over the other HER2-containing ADCs and Herceptin as a whole. It's a Herceptin-based molecule that was incrementally optimized in a very beautiful fashion that gave exquisite activity. But it was an incremental improvement, right? Cabozantinib, another good example from our labs that we made, that we developed, that Toni, thank God, was involved very early on and really made into the global blockbuster that it is today, is a VEGFR targeting TKI that had a different angle in terms of driving resistance, okay? And that's helping now tens of thousands of patients every day because of that simple incremental modification. But it all comes down to having the conviction, having the resources, and having the, in some ways, stubbornness to run large, global, randomized pivotal trials, not non-randomized, single institution phase 1Bs, but do the right work to get the right data that people like Toni need to get the job done.
Thank you, Najat. Let me tell you also who Toni is. So I'm a bit different. I may need an introduction. I'm a medical oncologist, and I see patients every week. Actually, I just came back from clinic. I work at Dana-Farber. I'm a genitourinary oncologist. I'm a physician scientist. I have a lab. I work all the time with industry, but I haven't worked in industry. I co-founded, companies. My CEO is here, Rehan Verji, a liquid biopsy company. And I have a different take. My take is really, different. Let me tell you what I'm positive about. The 3 or 4 things I'm positive. There is no room for negativity. One is clinical trials have to change. I have no idea why a randomized phase 3 costs at least 200, 250 million. When I started my career in 2007, it used to be 20 million. So what's going on? One of the last clinical trial I ran, do you know how many CROs were involved in general? Different CROs, 8. 1 for PK, 1 for imaging, 1 for biomarker, 1 for SUSAR. So $300 million, one company have all the drugs. $300 million. I think we need to sit down and see, and this trial was positive but didn't lead to registration. What is happening? This cannot continue. Cannot continue unless, like Belgium, you all accept that your taxes should be 60%. Well, at the end of the day, my sister lived there, what's more important than health? Make it 75%. But no, none of you are going to accept that. So you need to change that. One. Second, clinical trial control arm worry me a lot. So you have 2 drugs, let's say 1 versus 2 drugs. And you started the trial in 2024, gonna be read in 2027, but the OS endpoint gonna be 2028. The standard of care and the survival of patient change. And it didn't change because of subsequent therapies only. It did change because in general, humanity gets better. These patients have access to hospital. The support system is better, on and on and on. How can you adjust for the control arm? You cannot. So what we started at Dana-Farber and many other places, we're working closely with Flatiron, and I'm just reviewing the metastatic breast cancer because we have a large data set. We're looking at synthetic data using AI. And you will be surprised how things change. Can we do a phase 3 trial, monotherapy, where concomitantly there is a 1,000-patient control arm that evolved from real-world data? I mean, why is that? It's going to be cheaper and more believable. So that's second. Third, what I'm positive...
You should talk to the new head of CBER about that.
Oh, yeah. Well, that was 2 hours ago. Good luck. Look, if any of you want a job at Dana-Farber, if you're a clinician, you need to learn EPIC. If you survive EPIC, you find a job. The third thing I'm positive about is that today in the US in healthcare, it's a time of major crisis at every level. And because it's a major time of crisis, it's a time for a tipping point. And that's what Malcolm Gladwell talked about in his first book and second book. Do you want to change? This is the time, because this is the time of crisis. And you all are in the industry. I'm only an academician. I need to finish my notes tonight. So I'm positive because of the negativity that's there. And final one, Najat told me not to say it, but I have to. So I told Najat not to start by me. It's like, Toni, don't worry. I'm going to start by the least good looking and go to the best good looking.
I never said that. I did not say that.
So,
Sam, that's yours. And guess what? Lori Glimcher is not my CEO anymore, so you can't talk to her.
I have to say that as I think about clinical trials, and I've done a lot, I don't love the idea of worrying about p-values. Now, I say that not glibly.
At your own risk, right?
I know. It is at a risk. But quite often we get drugs approved, not because they are statistically significant data that we're getting, but because they're medically important. And we have to start thinking more about, and we do think about p-values because we have to, but we have to think about drugs as being medically important, scientifically important. And then the data's different. It doesn't give you a p-value because, like the famous Genentech TPA streptokinase trial, where 3 patients either way drove the data, but rather because it's obvious that one drug is working so well and your control arm is irrelevant. And while incremental approaches, as you said about in HER2 and others, and even when we were doing work on Erbitux or Cyramza, incremental value is important. I learned that from my colleagues at Eli Lilly. It's important to do incremental things that are of incremental benefit as well, but it's really important when you change outcome for patients in a huge way and create a new paradigm that exists for how you move forward in therapy.
I also want to come in on the p-value discussion. My plan was to disagree with Sam, actually, but I cannot, actually, because I totally agree with that. I think we really need to think through how we think about statistics based on the phase of the clinical trial. And the reality is that most medicines, investigational medicines, are actually not transformative and should be discontinued as soon as possible in early development. So we can actually focus on medicines that are more likely not only to achieve statistical significance, but that will really transform the life of patients. And you will find out in early development, if you do experimental medicine clinical trials that are well designed, high density of data, where you capture the whole patient experience, where you look at all the relevant biomarkers defined as broad as possible, including imaging, including soluble biomarkers, genetic biomarkers, what have you. And you will find out, actually, forgetting about the p-value, if you do such a small randomized clinical trial, that you can apply a different test, which I call the BOT, which is the bloody obvious test. You need to see complete separation. And I think there will be an increasing value of using AI to do this. But until we have this in place, we need to use RI, which is real intelligence. Right? This is a judgment call. And I've been doing this for decades, I have to admit, in academia, as global head of development of GSK, and now in biotech companies. I don't remember any case where it was not completely obvious, based on a well-designed phase 1b/2a experimental medicine clinical trial, whether a medicine was completely not going to work or it was going to work. And you need to stop that. And I think this is one of the reasons why drug development is so expensive, that we continue to try to get medicines to the next phase if there's a small signal, and we are wasting a lot of resources there.
That was going to be one of my next questions, because, and you did not see my questions, I'm impressed. And we can model you. You can be one of the ways we model our next AI algorithm for picking up good signals. But in all seriousness, there are incremental medicines that are on the market. So the question is, how do you pick up what leads to true differentiation earlier? There were examples on AI. There were examples on self-good-lookingness. There is also examples on just wisdom. Yes, absolutely. So I'll maybe start with Mike. Thoughts?
Well, it's a really good question, and certainly in the oncology realm, the challenge is how do you separate a signal in phase 1b, which we all know can be very, very highly irregular based upon small number of centers, patient selection. I'm going to enroll the best patients because I believe in this, right? So there's a bias there versus a large global randomized pivotal trial where all those factors, they're not completely gone, but they're certainly minimized, right? So, you know, I think about probably the best example where the industry was fooled by early non-randomized data was IDO, right? Back, you know, 10 years ago, right? Where there was a very strong signal by the Insight group that drove a big collaboration with Merck, which drove, I think, you know, 6 or 7 simultaneous pivotal trials because the phase 1b data was so compelling against what was perceived to be synthetic controls. You normally see X with a PD-1 by itself. IDO plus X gave you something different. Now, of course, confidence intervals are wide in these kinds of situations. It's easy to be, you know, to want to believe, right? Statistics are the ultimate arbiter of what's real and what's not. So, Sam, I mean, you brought up TPA. That was 40 years ago. Okay, I was still in grad school then, okay? I can't think of an example in the last 25 years in oncology where a p-value or statistical significance drove a lack of that in a solid tumor drove approval. I just can't. And certainly nowadays...
How about Tarceva and pancreatic cancer?
And that was a 1-week benefit.
So
that was...
Okay,
by the way, one example. Thanks, Dave. One example out of 1,000. Good job. Okay. So
there are more.
Okay, can you give me 10 more?
Steve,
okay.
I'm going to protect you here.
Steve, yeah. So
the point is
wait,
that there are more
Sam,
than
I'm
that.
not done. Okay, let me just finish. It's super important that we just acknowledge science. Look, I'm a scientist. I was a chemist decades ago, so I'm involved in the discovery of cabozantinib. You know, lots of drugs in the clinic and on the market now. The science is fantastic. We have to prove that it works, and the way you do that is by running the right trials and actually moving the needle for patients. Right? Go ahead, please.
Absolutely, with a caveat. There's always a caveat. The point is, it's very different. You can look at a patient, and there have been lots of examples. I know that I did it. Our job is to connect dots and to know what's going to work, to be prescient about the future. If we don't do that correctly, we fail. And while clinical trials and correct clinical trials are critically important, and if you're going to do a big randomized trial, p-values do become important. But more importantly, as I said, things have to be medically important, and you can prove that. And in cancer, we have, with trials of 110 people or trials of 50 people, you can prove it if you've got a drug that's a game changer. You can't prove it with something that may have a slight incremental benefit. It just won't work.
Maybe I can offer a dialectic solution to this discussion. I mentioned briefly the value of traditional statistics and p-values depends on the stage of development. And of course, if you do a registration clinical trial, you need to hit your p-value and your primary endpoints and your secondary endpoints. I think the misunderstanding about early experimental medicine clinical trials is that it should not be focused on positive results. It should be about the negative predictive value. You want to stop all the programs that are not going to be transformative. And I'm not aware of any example of a medicine that showed marginal efficacy and biomarker activity in an early trial that became a huge success in terms of transforming the life of patients and creating huge commercial success. I'll give an example. I don't want to offend anyone who works for Amgen. But I practice reverse rheumatology at this moment. I focus on oncology, but I'm a rheumatologist. And I tested all these new medicines, including the first TNF blockers at the time, actually, in the late 90s and beyond. I remember the first results that we analyzed for anakinra. Right? And I did the serial biopsies, and I was an advisor to Amgen. I said, I don't think this is going to be a transformative medicine for rheumatoid arthritis, because there are effects, but they don't blow you away like the molecule CA2 did at the time, later known as infliximab. Right? And it was developed. and it hit the statistical p-value. And it was commercialized, but actually I don't know any rheumatologist who's ever prescribed anakinra for rheumatoid arthritis. Now it turns out that it worked in periodic fever syndrome, so there was another future. And it's interesting. But
Amgen sold the drug because it wasn't working for them.
Exactly, but it was exactly. So this is an example. There are many. So I think we need to rethink about what do we try to achieve at each stage of drug development to become more efficient.
You know, Paul, just on that, and then I'm going to go to Toni. There is something around, we can measure more things today. Real time, right? ctDNA, transcriptomics, et cetera, and also with some of the examples with AI, you can actually have different synthetic control arms. So maybe back to you, Toni, like, what are those things that we can do more? And if you had any more rebuttals from Mike, you know, Sam, or anybody else, please.
Well, I think we need to go back to the basics. For example, there is nothing that predicts better success than single agent activity. What's the response rate to IDO inhibitor single agent, the inside compound? 0%. Now, how many times we've done that experiment? Almost nothing. We put it from PD-1. And it was a time, this is why I think this time is actually very good. I'll tell you why. It was time where there was a lot of money, a lot of cash. Let's take a chance. Zero activity. And even that, there was no attempt to create a biomarker for IDO inhibitor, because also the response rate was 0. So how did this take it? Because there was nothing else exciting to combine with PD-1. There was a lot of money. There isn't a lot of money today. This experiment will not get repeated. Why? Human stupidity is still the same. It hasn't changed. But there is less money. I'm sorry, this is what it is. I'm not going to put a patient on that combination. Now, the lucky thing is that toxicity weren't worse. Great, that's the thing. So I'm going to keep short because Sam needs to talk.
All right, on that very exciting note, but no, it is important to track these things earlier, and then to your point, put resources and be good stewards of capital. On that note, I'm going to wrap up and make sure we have time for some Q&A. Let's see. What about Rehan? Yes. You have a loud, booming voice.
So first panel, we're doing questions. I thought we'd given up on that today.
Oh, no, I stick to the rule. Haroon is very strict.
So I do have a question as we're closing, and I want to pick up on something that Toni said, which is just the cost and the time associated with clinical trials. And the fact that it's ultimately maybe costing us in terms of the number of important medicines that we can actually advance and do the right experiments for. So if you had one request, or you can make it two, Sam, for the FDA, you know, as they're starting to think carefully and re-examine some of the important regulations, what would be your ask so that we can get to some of that early phase 1B or even phase 3 data sooner?
You know, for me, again, speaking about something that's medically important, I'd go to the FDA and make sure that they understood. They have two goals at the FDA. One is safety, and that's a big deal. So we want our drugs to be safe. We don't want our drugs, and cancer drugs are easier than drugs for obesity or whatever. But we want our drugs to be safe. The next question is the question for the FDA to look at. We want our drugs to work. We want them to be efficacious. And when we do that, we can do it different ways. I'd request that the FDA do what they're already starting to talk about and begin to look at our phase 2 data, real phase 2 data, and say, does this drug look like it works? If it does, and we are beginning a registration trial, approve the drug temporarily, let the trials finish, start selling the drug. If physicians think that a drug's working, they're going to make sure that their patients get it. And payers are going to make sure that they reimburse. And we've seen that in many situations, and I'd request that the FDA started doing that, because it would start allowing biotech companies, start allowing pharma companies to do trials more efficiently, and make sure that those trials don't break the bank.
And maybe for the post-marketing commitments that are invariably there, I think you mentioned this, going back to EPIC, You can actually monitor some of the data. Not perfect, but you can monitor some of it.
But
Any
better
other
than
comments?
having nothing.
What?
But better than having nothing.
Yeah, exactly. We have time for one more question.
There's been a lot of focus in your discussion about p-values. Okay. You had a lot of discussion about determining efficacy and the role, or maybe skepticism about p-values and whether a drug was worth developing. But you know, many drugs are lost for safety issues, especially in phase 3. And those drugs might actually have had very high efficacy signals, but there's the possibility of the play of chance in a randomized trial generating something that looks like a signal, and it may or may not be a signal. So I wonder if you could just spend a little time on examining the role of the p-value in safety versus efficacy.
Mike, do you want to take that first? And then Steve.
Yeah, I guess I mean, Toni could probably speak this better than I can, but in the context of the totality of data, it's important to look at the risk-benefit, right? So the stats don't often apply to the individual safety components because you're looking at potentially hundreds, if not more, different adverse events in oncology. I think the issue is more around, do you have the right dose that gives you the right therapeutic risk-benefit ratio to be able to make that work from an efficacy point of view. So Toni, do you want to speak to that?
There is no p-value with safety. That doesn't exist. You're going to look at death on study, you know, in both arms. You're going to have a semi-objective way to look at. You're going to see if patients, how much they have dose reduction, complete dose discontinuation, death. We're going to look at the specific areas, nausea, vomiting for a few days. That is not as even neutropenia compared to hospitalization, even though the patient maybe can't be at home. That's why they're hospitalized, by the way, or things like heart and neuro. So the toxicity for p-value really doesn't apply. It's an efficacy thing. And I agree on clinically relevant. Clinically relevant also is in the eyes of the beholder. Every disease at every stage for every trial are different what's clinically relevant. A clinically relevant 1 week in prostate cancer is not like pancreatic cancer where patients and doctors and family members just hold into that 1 week survival advantage and that p-value.
Yeah, I might kind of just, I think we've mistaken a couple of concepts. One is proving something in its place in the marketplace versus understanding, do you have something that actually has a biologic impact? I think that's kind of, and that's probably true both in safety and efficacy, right? So you're signal seeking in phase 1B on the efficacy side. You want to see everything that's internally consistent. And if you don't, and you don't have single agent responses, the probability it works is very, very low. And I would say the same thing in a randomized trial for safety, right? You're not really looking for a p-value, you're signal seeking. And so when you see a signal and it's consistent with the mechanism and or it's a really bad outcome, you have to take it seriously. But if it's inconsistent and it isn't, and maybe it's a rash and you have a survival benefit, it's worth studying. It might be worth studying a broader set, but you're really just finding a signal that you don't necessarily want to react to.
Toni said something very important because the first thing he mentioned was looking at deaths. You did, Toni. And what's very interesting is in some clinical trials, you'll see in the experimental arm, 2 patients will die. And physicians note that, and they live with that. And they think if there's a competitive drug that didn't kill those 2 patients, it's going to be used differently than another drug. It's very important because it isn't p-values. It's just looking at what you see as side effects.
I'll just add, the problem is that you need much larger numbers of patients that have been treated to really get an idea about safety. And therefore, I think a more creative way of having post-marketing requirements to really monitor patients is the answer there.
On that note, I am right on time. Thank you, everyone. Just a big round of applause. All right.